So let's look at how a root grows as an example of how primary growth and cellular differentiation occur. Let's examine the structure of the root, starting with the tip. The outermost cells on the tip of the root are actually non-living. Even though these cells are dead, they still have an important function. They provide a buffer between the living and growing portions of the root and the soil. The pointy shape of this root tip helps to push soil aside as the root grows deeper and deeper in search of water. Just behind the non-living cells of the root cap is a zone of active cell division. Okay, so it's this region that's actually the apical meristem of this root. We can easily see the evidence of mitotic division if we look at the slide that's been stained. Keep in mind that chromosomes are only visible as dark structures in cells that are actively undergoing mitosis. Okay, and that's because the chromosomes are condensed during mitosis and the DNA is not visible. It's loosely strewn about the cell as chromatin during interphase that occurs in between mitotic divisions. So in this stained root here, we see that most of the mitosis is indeed going on in that region, located just a little bit behind the root tip. Okay, so when we look at these cells here, we can see they're very small because they're rapidly dividing. The stem cells, those that retain the ability to divide, remain in this region. And from every cell division, one of the two daughter cells migrates upward and begins the process of differentiation, while one of the other cells remains in the apical meristem and retains the ability to divide. The differentiating cell is going to grow longer and longer, and we see the zone of elongation occurring. So even though the division of cells is important in root growth, it's actually the elongation of those cells that's most primarily responsible for the increase in root length. Notice the shading on this root here. The color coding designates each cell as belonging to the dermal, the ground, or the vascular tissue system. And the darkening of these colors as we move upward in the root indicates the greater and greater degree of cellular specialization that's occurring as a cell gets farther and farther away from the growing root tip. So signals from surrounding cells cue the cell that it's time to differentiate. It's time for some genes in the cell to be turned on and others to be turned off. The cells in the center always become the vascular tissue of the root. The cells on the outside of that become the ground tissue, and a single layer on the outside become the dermal tissue. Some of the cells within the dermal tissue develop into root hair cells. Depending upon the amount of water available in the environment, A root in a dry environment would need to grow more root hairs in order to take up enough water, and the density of root hairs in the surrounding cells. In the previous picture, we saw a lengthwise section of the root. If we were to cut a root crosswise, within the zone of differentiation, in which the different tissue types have already fully formed, we might see something like this. We have an example here on the left of a typical eudicot root, and an example on the right of a typical monocot root. Although the exact arrangement of cells will depend upon the species, these two pictures show some generalizable similarities and differences between eudicots and monocots. Okay, some of the traits that these have in common are an epidermis on the outside and a vascular cylinder containing both xylem and phloem on the inside, with a layer of cortex in between. And then we always have a layer called the endodermis. Okay, remember that endo means inside of, whereas epi means on the outside of. The endodermis surrounds the vascular cylinder, providing a buffer between it and the surrounding tissue. But notice that the arrangement of vascular tissue within the vascular cylinder differs between the eudicot and the monocot. This X-shaped structure that contains the xylem is typical of eudicots, with the phloem found around the outside of it. Most monocots have smaller scattered bundles of xylem and phloem within the vascular cylinder. Also note the additional parenchyma cells found in the center of the monocot, but not in the eudicot. So the vascular tissue at the center of the root has the important function of drawing water up from the ground into the rest of the plant. The ground tissue surrounding the vascular tissue is mostly parenchyma cells. Okay, and can you remember what the primary function of parenchyma is? Okay, it's the metabolic function. We don't have photosynthesis going on in roots, since they're not exposed to light, but we still have to have metabolism occurring in all parts of the plant. So the parenchyma carries out the metabolic functions of the root. The endodermis is an important thin layer that forms the innermost edge of the cortex. And it has an important function in regulating or determining which substances pass through the ground tissue from the soil into the vascular cylinder. So it's important in deciding which substances the plant needs from the soil and which ones it wants to avoid. Because once inside the vascular cylinder, a substance is then going to be distributed throughout the plant. Just inside of the endodermis, okay, remember that the endodermis is the innermost layer of the cortex or ground tissue. Just inside of the endodermis is the outermost layer of the vascular cylinder. And we call this thin layer of cells the pericycle. And it's The pericycle is the region from which lateral roots can arise from existing roots. Okay, these are roots that grow out from the side, and here we can see a bud of a lateral root beginning to form and enlarging. We can start to see a new apical meristem forming, and the zones of cellular differentiation beginning to form as the root grows outward. So the density of these lateral roots and the exact placement of them is going to be influenced by environmental factors that stimulate root growth, such as the presence or the scarcity of water in the area. So keep in mind that shoots grow more or less in the same way as roots. Okay, we don't have quite as many dead cells at the end of a shoot as we do at the end of a root, but we still have the same pattern of intense cellular division in the apical meristem and then increased cellular differentiation as we move away from that meristem. On a shoot, we have additional structures forming called leaf primordia. These are the beginnings of new leaves forming along the sides of an apical meristem. So just like in the root, the position of the cells relative to the apical meristem determine what type of cell each one will differentiate into, with the outermost layers of cells forming these leaves and the innermost cells elongating and forming more of the length of the shoot. Remember that plants have apical buds at the end of shoots and axillary buds off to the side. So we can see in this diagram there's only one apical bud at the end of each major branch. And these axillary buds are found at the nodes at the base of the junctions between the stem and the leaves. So growth is actually going to be the most active at an apical bud because of a phenomenon called apical dominance. And apical dominance occurs when growth at an apical meristem, when growth at an apical bud actually releases hormones that inhibit the growth of lateral buds that are nearby that region on the plant. Okay, so this means that under normal conditions, the plant's going to primarily produce leaves in an upward direction. But if something happens to that apical bud, let's say that a hungry herbivore comes by and eats these tender, juicy, newly developing leaves, that apical bud no longer suppresses the lateral buds, and then we would probably get several new branches, maybe here, here, and here, growing out from the side of the plant. Okay, so typically the loss of an apical bud results in the stimulation of growth in two or more axillary buds. So probably at some point in the history of this plant, damage to an apical bud resulted in an axillary bud causing this branch to grow off the side of the plant. Keep in mind that the leaves growing off the main stem, such as this one, and this one, were formed at the time that this apical bud was here on its way growing upward to this point. So it's knowledge of the apical dominance that explains why when gardeners pinch off a bud of a flower or shrub, they tend to get more bushy lateral growth and the stimulation of additional leaves that they would not have seen formed if they hadn't pruned back the leaves from the apical bud. So gardeners can get a plant to grow in the desired shape by understanding the growth patterns of plants. Okay, the tissues that are found in stems are an extension of the tissues found in roots. Even though these tissues are continuous, from the apical meristem at one root tip up through the apical meristem at the tip of a shoot, often the arrangement of cells within these tissues differs somewhat between roots and shoots. And as you might expect, there are some general differences between monocots and eudicots. These pictures here represent a common pattern that we see in monocots, with the vascular bundles scattered throughout the ground tissue of the stem. And eudicots tend to have larger aggregations of vascular tissue, forming a ring around the outside of the stem. Now, both of these pictures here are of non-woody species, and we tend to get different patterns in woody species, and we'll see that in a moment as we examine the trunk of a tree and look at the annual rings.